Cancer presently remains the leading cause of death in Canada, as an estimated 2 in 5 Canadians will receive a diagnosis in their lifetime, and unfortunately, about 1 in 4 will eventually pass away. Glioblastoma, or GBM, is an aggressive brain tumor and is also one of the most persistent human cancers. Given the gravity of its impact on the lives it intrudes, it's worth taking a look at the intricacies involved in further propelling this disease, with the hopes of working towards a more complete understanding that can help effectively guide future treatments. GBM growth is derived from stem cell-like cells. Bone morphogenic protein, or BMP signaling, triggers the cell cycle exit and astrocytic differentiation of these GBM stem cells, and therefore may have therapeutic value. GBM is characterized by an extensive hypoxic environment that triggers invasive cancer cell phenotypes, and this tumor invasion is a major contributor to chemoresistance. Over the past couple of months, we've been focusing on understanding whether or not hypoxic conditions influence BMP's ability to induce differentiation across various GBM stem cell lines. Our experimental workflow has involved preparing for growing our cells by first appropriately coating our flasks to allow for effective adhesion of our GSC culture, which we then grow in growth factor media. Depending on the experiment, we then proceeded to seed our cell lines and then incubated it in normoxic conditions for 3 days. We then added our differentiation media, which does not consist of growth factors but includes BMP4, and we also mimicked hypoxic conditions with a histone lysine demethylase inhibitor GSKJ4. We then respectively grew the treatments in either hypoxic condition of 0.2% oxygen or in normoxia for 3 days. At the end point of our experiment, we have 3 potential assays. The first one is our visual InQSight data, which continuously scanned our plates over 8 days and produced a graph like the one on the bottom right, presenting the decrease in confluency seen in the G895 stem cell line as a result of BMP treatment. After harvesting our cells, we then proceeded to carry out either Western blots for protein visualization or qPCR for gene expression. The leftmost graph in the results section illustrates the expression of the astrocytic marker GFAP in 6 treatments in the G885 stem cell line, in which BMP caused an over 10,000-fold increase in GFAP expression in normoxia. With a similar increase in hypoxic conditions, and a similar induction pattern was seen in the GSKJ14 treatment as well. And these are all findings similar to those across the other cell lines we've looked at so far as well. So, although it seems like BMP does induce differentiation across GBM stem cell lines, hypoxic conditions hinder its ability to do so. So in the future, it might be valuable to test for the undesirable latent ability to re-enter the cell cycle by testing for responsiveness to re-exposure to growth factors. But overall, the data that we're continuing to collect as of right now is helping us characterize hypoxia's effects on BMP-induced differentiation, and will help further illustrate any potential caveats in the use of BMP in GBM differentiation therapy.